number 6 about visualization of predicted structures. As I was explaining that UCSF Chimera is a big suite that can perform many experiments like minimization of protein, analysis, simulation, structure prediction, structure visualization, RMSD analysis, and many more. So we just use this software for structure visualization. Visualization through different options. First of all, file option. First you can open a three-dimensional structure in Chimera, or you can just write the PDB code like PDB accession number, and then you have the three-dimensional structure. You can save the session status, like if you just perform many experiments, and as you know when you just close the UCSF Chimera or exit from the software, you cannot get the same data back. You will be on the first step. So you can save the session in python.py file, and after that when you will open the .py file in Chimera, you will be on the same position where you left. And here we just open the RAS-F2 protein from the open option, file option, and then we just click on open. You can save the image by save as option. Here you can have a big information about that image setup, preferences, and how to save the image, how to change the length of the image and pixels of the image and DPI of the image, how to make it more beautiful. After that, you have the option of select option, where you can select the backbone of the protein, ions if there is any ion, or if there is any ligand, if there is any heme molecule cofactor, you can select directly. There are two ways from the bar windows that is already shown in figure number 69, or otherwise you can just place your cursor or the specific position, press the control button from the keyboard and click there. You can highlight that portion and select the portion. You can also select the nucleic acid protein, main protein, and the ligand protein, and the backbone of the protein, and the solvent of the protein, coils of the protein, beta sheets of the protein, and other different types of parameters you have in this. Then you have residues, the option of residue in the select option icon, in which you have all the residues present in the specific protein you are visualizing. And in this protein RAS-F2, we don't have in actual any of cofactor or prosthetic group attached. Otherwise, here if you can see the cursor, here we have the standard amino acids A, B, C, D, E, F, and here we don't have any prosthetic group or heme or water molecules or ions. If it was attached with the protein, then you can see that icons here, like heme molecule, or maybe zinc molecule, or maybe iron ion, or ligand, or anything else you can see here. After that, we can see the chemistry of the protein, that how many carbon atoms here, nitrogen atoms, oxygen atoms, and chlorine atoms, and all that material you can see easily the chemistry of the protein from here. Then we have another icon, that is the third option here, action, in which you have items and bond. If you just select the action and go on the items and bond, then you have a new window appear, and here if you just select on the show, you can have all the items and bonds. Here you can see in figure number 72. And if you just specifically select some items and show the items and bonds of that specific residues, suppose you just select all the alanines or just all the prolines from the protein, and you just want to show the items and bonds of only proline or only from alanine, you just select all the alanines and show the items and bonds. You will have only the items and bonds of alanine or proline or any you want to express. And if you just select it, like I saw many people just generated different types of figures. So if you select the proline or the beta plated sheets and you want to change the shape of the beta plated sheet, now you have the options here. If you can see the cursor, you can change it into sticks, ball and sticks, sphere shape, wire shape, and you can increase the width of the wire to some extent according to the experiment and according to your wishes or the demand of the figure. After that, we have the option of ribbons. Here we can show the ribbons or we can hide the ribbons by selecting the specific ribbons or all the ribbons of the protein, or you can also flatten the selected ribbons, or make it bit edged, or make it round by the different options. you can see here in figure number 73. Then you have another option in Chimera, you can see in figure number 74, showing surface, in which you just have to select the whole protein first, and then you just go to the option of surface and show the surface. You will have the complete surface of the protein, and you can change the surface into mesh, into dotted, or into solid plasma type shape. Or you can also use the transparency option to just reduce or increase the transparency of the surface. Like here you can see in this figure number 74, you just have 80% selection for the transparency. Now you can see the ribbons and alpha helices and beta plated sheet. If there is 0% transparency, you cannot see anything, and if there is 100% transparency, surface गायब हो जाएगी. 0% पे surface 100% शो होगी. 80% पे आप देखें surface कितनी dim है figure number 74 में. तो 100% में ये surface बिल्कुल गायब हो जाएगी. आप coloring the secondary structure elements. It's a very big tool, and there is so many options in UCSF Chimera. And trust me, if you have a good hands on it, you can go much far in the visualization of proteins, analysis of proteins, generating diagrams for the protein. So in figure number 75, we have the option of, so you can color, you just select the specific items or bonds by different parameters through the clicking, as we just discussed above, or just use the control option, and you can select specific items or specific bonds, and then you can give them a specific color what you want: black, white, yellow, red, orange, ABC. After that, we have the option of label residues, and in label residues you just can select any of the residue, like you just selected for coloring the residue. You can label the residue, just select the residue and open the label option, and you can label the residue. You can label the residue with 3-letter code of amino acid. You can label the residue by 1-letter code of amino acid with the number of that amino acid. Like serine 14, you can label it like SER14, or you can also label it like S14. If you have many residues, and when you will show the 3-letter code of the amino acids, there will be a mess on the figure. In that case, you can just show the of amino acid, 1-letter code of amino acid with the number. And if you have less amount of or less number of amino acids, you can show the 3-letter code of amino acid. Depends on the need of the figure or your choice. Here you can see in figure number 76, we have arginine 163, histidine 167, isoleucine 171, and arginine 166, arginine 165, glycine 173, and histidine 174. We just label that because RAS-F2 has this binding pocket in actual that we just showed, I think so. Okay, now we will be on preset options. In preset options, you have 3 or 4 different types of styles, and you also have the tools or sequence and different types of icons in which you can adjust the light adjustment option, in which you can adjust the light. There is a torch. When you just open that icon, you have UCSF Chimera in your laptops. You can just open any 3-dimensional structure and perform all these experiments for your learning. It's much better for you if you will perform this experiment and practice on this. Just upload a 3-dimensional structure and go to the preset option. You have light adjustment option. There will be a torch. You put your cursor, place your cursor on the torch and click and drag the torch. You will see the light will move with the torch on your protein that you uploaded. Then you have interacting residue analysis. You can perform it. The analysis, I remember you guys just know about that, but I am going to explain it again. First, you have to select the structure analysis, or select the specific residues, or maybe select the ligand or the interacting protein, depends on the experiment. Then you have to find the clashes and contact. You just have to click the option of clashes and contact, and you will have the contacts with the amino acids with one another. Or maybe, okay, we have this figure number 78 for changing the intensity of light. Here we have the torch like option, you can see in this figure, and when you just move it, you can change the light from this structure. Please focus on the cursor of the mouse from where I just want to explain. I try to explain. After that, you have the option of designate. After find clashes and contact from this window, you have the option of designate. Here you have many parameters. Your contacts should be -0.4 Armstrong, and your clashes will be 0.0. After that, you have four options here. First one is the select option. In the second one, you can color with the red. In the third one, I unchecked these options in actual, but you can use these options. I usually use the first one and the fourth one, but I am just explaining all the options for you if you want to use that. In the third option, draw pseudo connections in the yellow color, and the width of the interacting ligand or the residue of the protein should be 2 Armstrong. Then you have the yellow lines from that which amino acid is interacting with which amino acid. Like serine is interacting with leucine, there will be a yellow line between that, because usually students or the newcomer or the beginner don't know which amino acid is interacting with which amino acid. And if you will generate the log file, you can know it very easily. Or you can also find the distance between the two interacting residues in Armstrong. I hope and I believe in many papers you guys read about that in the tables, that serine is about 1.5 Armstrong far from leucine, and it has this strong affinity. So if you just use these options, you can find the distance for this yellow option, the third one. In the fourth one, you are using the overlap option, assign overlap attribute. In fifth one, you will have the information file that has the detailed information of the interacting residues, and in the sixth one, you will have the reply log file. Depends on your need. I usually check one and fourth one because I just have much experience in this, so I can understand. If you guys want to understand more, you just have to generate the log file and the information file for your much more learning. Okay, here we have this option that I already described to you, one letter code and code specifier. You can see here, maybe, okay, I can highlight it for you. Then the most important step we usually perform in Chimera is energy minimization. Why we minimize the energy? Energy minimization is the significant process to minimize the protein for further drug design studies. To minimize the protein, open the file, upload the file, use the tools option, structure editing, and minimize the structure. In the minimization steps, we usually form it at 1000 steepest descent steps and 1000 conjugate gradient steps to minimize the protein. Here we also have another mistake, F is small, it should be capital. Okay, so then you have to minimize the three-dimensional predicted structure to make it more reliable. You can see here, you just have to write 1000 in conjugate gradient steps in Armstrong, and you also have to write 1000 in steepest descent steps in that bar. So it will be much good for you. It will hang your laptop if your laptops are not much good, but you don't need to close the software. You just have to wait it. It also take much time, depends on the number of amino acids in the protein. Like if you have 200 amino acids in the protein, maybe it will take 5 hours, and if you have 500 amino acids in the protein, it will take much longer for that. It's limitation of structure visualization tool. UCSF Chimera takes much time to save in Pauvre images, and it also takes much time when you are going to minimize the protein. After that, we have this table, and as we use one tool in this chapter, so there is one tool in this table. It is freely available tool. Description is here, workflow is here, URL is here.